Mediators of inflammation. The mediators of inflammation are the substances that initiate and regulate the inflammatory reactions. Many mediators have been identified and targeted therapeutically to limit the inflammation. In this discussion, we review their shared properties and their general principles of the production and action. The most important mediator of acute inflammation are vasoactive amine, lipid products like prostaglandins and leukotrienes, cytokines, including chemokines, and product of complement activities. Principal mediators of inflammation: histamine, prostaglandin, leukotriene, cytokines, TNF, IL-1 and 6, chemokines, platelet activating factor, complement and kinin. Histamine from mast cell, basophil and platelets. Action is vasodilation, increased vascular permeability and endothelial activation. Prostaglandins from mast cell and leukocytes leads to vasodilation and pain and fever. Leukotriene from mast cells and leukocytes leads to increased vascular permeability, chemotaxis, leukocyte adhesion and activation. Cytokines TNF, IL-1 and 6, macrophages, endothelial cells and mast cell. Local endothelial activation, expression of adhesion molecule, systemic fever, metabolic abnormalities and hypertension, shock. Chemokine leukocytes activated macrophage, chemotaxis, leukocyte activation. Platelet activating factor from leukocytes and mast cell, vasodilation, increased vascular permeability, leukocyte adhesion, chemotaxis, degranulation and oxidative burst. Complement from plasma produced in the liver, leukocyte chemotaxis and activation, direct target killing of membrane attacking complex and vasodilation, mast cell stimulation. Kinins plasma produced in the liver, increased vascular permeability, smooth muscle contraction, vasodilation and pain. This mediator induces various components of the inflammatory response, typically by distinct mechanisms, which is why inhibiting each has been therapeutically beneficial. However, there is also some overlapping redundancy in the actions of mediators. Mediators are either secreted by cells or generated from plasma proteins. Cell-derived mediators are normally sequestered in intracellular granules and can be rapidly secreted by granule exocytosis. Histamine in the mast cells or are synthesized de novo, for example, prostaglandin, leukotrienes, and cytokines in response to stimulus. The major cell type that produce the mediators of acute inflammation are the sentinels that detect the invaders and damage in the tissue. That the macrophages, dendritic cells and mast cells, but platelets and neutrophil, endothelial cells and most other epithelia can also be induced to elaborate some of the mediators. Plasma-derived mediators. For example, complement proteins are produced mainly in the liver and are present in the circulation as inactive precursor that may be activated usually by the series of proteolytic cleavage to acquire their biological properties. Active mediators are produced only in response to various stimuli. These stimuli include microbial products and substances released from necrotic cells. Some of the stimuli trigger well-defined receptors and signal pathways described earlier, but when still do not know how other stimuli induce the secretion of mediators, for example, from the mast cells in response to cell injury or mechanical irritation. The usual requirement for the microbes or dead tissue as initiating stimulus ensures the inflammation is normally triggered or when or where there it is needed. Most of the mediators are short-lived. They quickly decay or are inactivated by enzymes, or they are otherwise scavenged or inhibited. There is thus a system of checks and balances that regulate the mediator action. These built-in control mechanisms are discussed with each class of mediators. One mediator can stimulate the release of other mediator. For instance, products of complement activation stimulate the release of histamine and cytokine. TNF acts on endothelial cells to stimulate the production of another cytokine, IL-1, and many chemokines. The secondary mediators may have the same action as the initial mediators, but may also have different and even opposing activities. Such cascades improve the mechanism for amplifying or in certain instances counteracting the initial action of mediators. We next discuss the important mediators of acute inflammation, focusing on their mechanism of action and roles in acute inflammation. Vasoactive amine, histamine and serotonin. The two major vasoactive amines are so named because they have important action on blood vessels are histamine and serotonin. They are stored as preformed molecules in the cells and are therefore among the first mediator to be released during inflammation. The richest source of histamine are the mast cells that are normally present in the connective tissue adjacent to the blood cells. It is also found in blood basophils and platelets. Histamine is stored in mast cell granules and is released by mast cell degranulation in response to variety of stimuli, including physical injury such as trauma, cold or heat, by unknown mechanism, binding of antibodies to mast cells, which underlies the immediate hypersensitivity reactions, and products of complement called as anaphylactosis C3A and C5A, described later. Antibodies are complement products, bind to specific receptor on mast cells and trigger the signaling pathway that induce rapid degranulation. In addition, leukocytes are thought to secrete some histamine releasing protein, but these have not been characterized. Neuropeptides, for example, substance P and cytokines IL-1 and 8, may also trigger the release of histamine. Histamine causes dilation of arterioles and increase the permeability of venules. Histamine is considered to be principal mediator of the immediate transient phase of increased vascular permeability, producing interendothelial gaps in venules As discussed earlier, its vasoactive effects are mediated mainly via binding to receptor called H1 receptor on microvascular endothelial cells. The antihistamine drugs that are commonly used to treat some antagonists that bind to and block the receptor. Histamine also causes contraction of some smooth muscles. Serotonin 5-hydroxytryptamine is a preformed vasoactive mediator present in vessels and certain neurocrine cells such as gastrointestinal tract and in mast cells in rodents but not in humans. Its primary function is as neurotransmitter in the gastrointestinal tract. It is also the vasoconstrictor, but the importance of its action in inflammation is unclear. Arachidonic acid metabolites, the lipid mediator prostaglandin and leukotrienes, are produced from arachidonic acid present in the membrane phospholipid and stimulate vascular and cellular reaction in acute inflammation. Arachidonic acid is 20-carbon polyunsaturated fatty acid. 5, 8, 11 and 14 eicosapentatetraenoic acid is derived from dietary source or by conversion from essential fatty acid linoleic acid. It does not occur free in the cells but is normally esterified in the membrane phospholipids. The mechanical, chemical, physical stimuli or other mediators like C5a release arachidonic acid from the membrane phospholipid through the action of cellular phospholipase, mainly phospholipase A2. The biochemical signals involved in the activation of phospholipase A2 include increase in cytoplasmic calcium and activation of various kinases in response to external stimuli. Amino acid derived mediators, also called as eicosanoids, because they are derived from 20-carbon fatty acid, are synthesized by two major classes of enzymes, cyclooxygenase, which generate prostaglandin, and lipoxygenase, which produce leukotrienes and lipoxins. So, cell membrane phospholipid steroid inhibits the phospholipase, whereas the phospholipase leads to the production of arachidonic acid. Other lipoxygenase produces HPETEs and HETEs. Cyclooxygenase and 5-lipoxygenase produce prostaglandin G2, then H2, then I2, which causes vasodilation, inhibits platelet accumulation. Thromboxane A2 causes vasoconstriction and promotes platelet aggregation. PGD2 and E2 causes dilation, increased vascular permeability. Cyclooxygenase is inhibited by COX-1 and 2. Inhibitors are aspirin, indomethacin, etc. Lipoxygenase 5-HPETE, which will further be converted to 5-HETE, which leads to chemotaxis, leukotriene B4, then leukotriene A4 from 5-HPETE, LTA4, leukotriene C4, D4 and E4. All of this leads to bronchospasm and increased vascular permeability. 12-lipoxygenase leads to formation of lipoxin A4 and B4, which inhibits inflammation. Production of arachidonic acid metabolites and their roles in inflammation. Note the enzymatic activities whose inhibition through pharmaceutical intervention blocks major pathways. COX 1 2 HETE HPETE. Eicosanoids bind to G protein coupled receptor on many cell types and mediate virtually every step of inflammation. From prostaglandin we will see in next video.